Genetics Matters, Episode 5, Avery's Disbelieving Proof. Welcome back to Genetics Matters. I'm Brad Goodner. In our last episode, we dealt with Frederick Griffith's seemingly senseless experiment concerning the respiratory pathogen Streptococcus pneumoniae. To summarize what we've learned so far, Streptococcus pneumoniae strains that can make an extracellular polysaccharide capsule and grow as smooth-looking colonies on a petri dish, so-called S strains, can kill mice. Streptococcus pneumoniae strains that cannot make an extracellular polysaccharide capsule and grow as rough-looking colonies on a petri dish, so-called R strains, cannot kill mice. If you heat kill cells of a S strain and inject them to a mouse, the mouse lives. If you inject millions of cells of an R strain into a mouse, the mouse lives. However, if you combine dead S cells with live R cells and then inject the mixture into mice, some of the mice will die. And the bacteria you find inside the dead mice are live S cells. Griffith's seemingly senseless experiment of combining two things that could not kill a mouse and getting something that could kill a mouse forced him to think about these cells and how they were becoming different. Something from the dead S cells changed some of the live R cells so that they now acted as S cells. Griffith called this phenomenon transformation. Unfortunately, he died in a German bombing raid on England and never got the chance to better understand this phenomenon, to identify his transforming principle. Across the Atlantic Ocean at Columbia University in New York City, Oswald Avery read Griffith's papers, but he wasn't convinced of transformation until someone in his own lab repeated Griffith's work and got the same results. Over the next few years, Avery's lab continued this line of research. They eventually came up with a more efficient transformation protocol so that they did not have to inject mice in order to find the rare R cells that have been transformed into S cells. Rather, there was a large enough percentage of transformed cells that they could simply look for the S colony morphology on petri dishes. Avery and coworkers then used biochemical techniques, both established and some of their own design, to break up the dead S cells and separate the cellular material into fractions that were enriched for particular biological molecules. They gave each fraction to live R cells and looked for transformation of some of the R cells into S cells. Remember that R cells cannot make the extracellular polysaccharide capsule normally seen in S cells. Maybe giving the dead S cell fraction rich in polysaccharides would allow some R cells to transform into S cells. However, polysaccharides, like most biological molecules, are made through reactions catalyzed by enzymes, and enzymes are almost always proteins. So maybe the dead S cell fraction rich in proteins can transform some R cells into S cells. To Avery's great surprise, neither of these two fractions that he thought were most likely to involved in transformation, polysaccharides or proteins, neither of them were able to change R cells into S cells. The only fractions that had transformation ability were fractions that contained DNA. Avery's lab figured out ways to further and further purify the DNA, and it was always the DNA fraction that could transform R cells into S cells. Avery realized he was onto something much bigger than just Streptococcus pneumoniae and its ability to cause disease. Transformation was about an organism becoming genetically different, and he had evidence that DNA was at the heart of the matter. But Avery and coworkers did not stop there. They also did removal experiments. Take an extract of dead S cells and treat it so as to degrade one particular type of biological macromolecule. For example, treat the S cell extract with proteases to degrade most proteins in the sample, and then ask if the treated extract could still transform R cells into S cells. Through these removal experiments, They found that removing proteins, polysaccharides, RNA, or lipids had no impact on transformation. However, degrading the DNA in the dead S cell extract abolished its ability to transform. Avery and his co-workers had proved through both positive and negative types of experiments that DNA was in fact the genetic material. DNA from dead S cells, when added to live R cells, could allow some of the R cells to change and become S cells. Their 1944 research paper is considered one of the all-time best because of the extensive controls done and the multiple lines of evidence, all pointing to the same surprising conclusion. This incredible amount of evidence stemmed from the fact that Avery found it hard to believe that DNA is in fact the genetic material. More than 50 years after the publication by Avery and co-workers, other researchers took those same Streptococcus pneumoniae strains and sequenced the region in their chromosome containing the genes involved in capsule polysaccharide biosynthesis. The original S strain had all the genes necessary to make capsule polysaccharide, but the R strains turned out to have a deletion in their chromosome that removed some of the genes encoding enzymes involved in the early steps of capsule polysaccharide biosynthesis. But how does the phenomenon of transformation allow R cells to become S cells? It turns out that Streptococcus is one of only a relative handful of genera in the domain bacteria that can naturally take up DNA from the outside environment and transport that DNA into its cytoplasm. Now, most of the time the DNA gets degraded, but sometimes it can get integrated into the host chromosome. If the entering DNA fragment shares high sequence identity to the host chromosome along two nearby stretches, then the entering DNA fragment can be recombined into the chromosome, taking the place of what was there previously. In this case, the S-type working copies of the genes can take the place of the deletion in the original host R cell. This is the way transformation works. Now we know the evidence for DNA as a genetic material, but how can such a seemingly simple polymer accomplish all that is needed? That answer awaits us in future episodes.